Hi everyone and welcome back. Today we're going to be talking about control and instrumentation in the pharmaceutical industry. There are two main components of a pharmaceutical product. The first is your API, or active pharmaceutical ingredient. This is the part of the drug that causes your desired pharmacological effect. Excipients, on the other hand, are inactive components. They may provide bulk, help to facilitate absorption, or be used for stability. These excipients can even help prevent the denaturation of your drug and help achieve a sustained release dosage. There are many ways in which you can set up your monitoring and control system in the pharmaceutical industry. The four most common ways are inline, whereby your sensor is placed directly into your process solution; online, where you divert the sample from your main manufacturing line and then return it after the monitoring process; at-line, this is when your sample is physically transferred from the process to a nearby analytical instrument; and finally, offline, which is where your sample is removed from your process entirely and taken for analysis at another location. This is what a standard liquid oral formulation manufacturing process looks like. So you begin with your sampling of the raw materials, where your raw materials are sent to your quality control laboratory, where it is analyzed to see if it passes the relevant tests. Once quality control has approved it for release, that is when the weighing and dispensing stage occurs. After the dispensing stage, manufacturing occurs. With liquid oral formulations, it's basically a big mixing process. Once the mixing processes are over and the entire manufacturing has come to a halt, again, you will be required to send a sample for quality control release. Quality control checks to see if the oral formulation passes according to USP or BP standards, after which they release it to the bottle washing and filling and sealing stage. After visual inspection, usually done through another quality check, the bottles are sent for labeling and packing, and after which it is released for dispatch. So if you look at this flowchart over here, you can see, as I mentioned before, the entire manufacturing process is basically just mixing. Majority of liquid formulations is basically comprised of a syrup. So you begin with your syrup preparation, to which water is added, and then with your active ingredients and your any excipients, they will be mixed in a vat for about 60 minutes, depending on the formulation in question. As I mentioned before, there are several quality control and quality assurance tests that need to be passed before the material is ready for dispatch. A process that is somewhat unique to the pharmaceutical industry is nitrogen purging. This is when nitrogen is shot into the bottle opening before the bottle is actually capped. And the reason that this is done is to eliminate the headspace in your filled bottles. And this is done to increase a product's shelf life, because any exposure to air can reduce the shelf life of a product. So allowing your nitrogen to replace the air actually allows you to enhance the longevity of your product and how long it can actually stay stable on your shelf. And obviously, checking for microbial load is an incredibly important part of your liquid oral formulation process, because liquid formulations have a greater danger of being carriers of bacterial load than solid formulations or dry formulations, which is why these formulations need to be tested for microbial contamination according to your USP and BP recommendations. So the commonly tested for bacterial strains include Staph, Salmonella, and E. coli, and a combined yeast and mold count is also performed. Looking at your tablets and capsules manufacturing process, you'll see quite a big difference when compared to your liquid oil formulation process. some would say it is more complicated and less straightforward than the liquid formulation process. Similarly to what we've discussed before, the tablet manufacturing process as well begins with your dispensing stage, where each ingredient has to be weighed and dispensed according to the batch size required. Next, sizing is done. This is done through the use of mills to reduce your average particle size. Having a homogeneous average particle size allows you better dosage uniformity, which is something that is analyzed in quality control laboratories. Next comes the mixing stage. To create a uniform and homogeneous powder mix, mixing equipment such as ribbon blenders or double cone blenders are used. Then we come up to granulation. There are actually two ways that granules are formed. That's the dry granulation process or the wet granulation process. Granulation is done in order to transform your fine powders into granules because granules can be compressed much more easily. So dry granulation is used for formulations which have excipients that are binders and have binding properties. This mixture is compressed into a tablet and then it is broken down into granules. Whereas through the wet granulation process, you add in a binder to create moist granules. The drying process needs to be carefully controlled because high temperatures can cause denaturing of your active ingredients. So this is usually done using a fluidized bed dryer and lubricant is added after the drying stage. Next comes tablet compression, or in the case that you are producing capsules, the encapsulation process. So the tableting process is performed using an automatic tablet press. You can see the picture down here. You've got lower and upper punches that push down in order to form a tablet. So your encapsulation machines function pretty similarly. You've got your capsules loaded into your encapsulation machines, into which your powder mix is poured into the capsules, which are then closed. And your tablets may later be coated in a sugar coating. As I mentioned before, temperature is an incredibly important parameter in pharmaceutical production, but relative humidity is also very important. This is because high moisture content in the air can result in agglomeration of your granule and powder mix and may even lead to API degradation. There are certain tableting machines that also require low relative humidity environments. In general, most pharmacopoeial guidelines advise for a relative humidity below 45%. Humidity is measured using hygrometers. You can have a manual hygrometer, like the one on the left, or a digital hygrometer, like the one on the right. Process validation is slowly becoming the convention in the pharmaceutical industry. Process validation is basically proving that a manufacturing process will consistently produce a product that meets all of our predetermined specifications. This is not a one-off exercise. It is done when beginning almost any batch, and it involves not only validating your product mixes or your quality control tests, but also validating all of the machinery used in the production of a certain batch and going through maintenance records relating to this machinery. You'll have noticed that we talk a lot about quality control when we talk about pharmaceutical industry processes. And that is because a lot of these pharmaceutical drugs are going to be used in very critical requirements. But the problem is when you are conducting your quality control tests offline, it can cause significant delays to the release of your pharmaceutical batches. So, in order to overcome this problem, we may be able to streamline the quality control process by performing all of our analysis inline instead of offline. So, this is a relatively newer concept called process analytical technology, PAT. So this comes as a response to your conventional pharmaceutical manufacturing moving from batch processes to continuous process mode, because for the longest time, it has been the convention to operate pharmaceutical manufacturing in batch mode, as people assumed that this was a more hygienic way to run, and it was much easier to control releases in line with your quality control tests. But switching to continuous mode can actually result in increasing your productivity and increasing the speed at which pharmaceutical batches are released for dispatch. Most analysis that we've been talking about so far in terms of quality control for pharmaceuticals can be performed with inline NIR analyzers, and this provides real-time release testing. So here is an example of how an NIR analyzer may be applied in the field. So here it has been used in food and beverage processing. NIR spectroscopy analysis is such a useful tool because it is a non-destructive technique, and it can be applied to liquids, solids, or even gases. It's preferable for food products because their water content can make it difficult to work with other analysis techniques, non-destructive analysis techniques. And it offers greater probing capabilities. You don't need to prepare your sample in any way, which is why it can be applied inline. NIR analysis can be used to detect moisture content, blend uniformity, and even coating uniformity. So it may well be the future of quality control in the pharmaceutical industry. Thank you for your attention. For more information on our process control solutions, please visit sasengineering.lk.